Hi, I am Ondřej Vysocký, and my summer of HPC project is called Developing the User Interface of the FFEA tool. This tool is developed by a computational biophysics group at the University of Leeds. FFEA means Fluctuating Finite Element Analysis, and it is a method for protein simulation. Most of proteins are known in their atomic structure, but simulation at the atomic level makes it highly computationally expensive and unfeasible. FFEA method discretizes the protein into tetrahedral mesh, and the dynamics due to thermal fluctuations are resolved through finite element analysis, which simplify the simulation. My project is called Developing the User Interface, but at the beginning there already was a simulation tool. It is called FFEA Viewer, and it is Python tkinter tool. These pictures represent features of the viewer: mesh structure, surface, and nodes IDs. Problem of the viewer is that it provides lack of features, and most of all, it is very slow when visualize a large dataset. But before we started, we thought about the problem and about possibilities to use something more common instead. I did some research and decided to convert output of the simulation to the VTK file format, which can be visualized, for example, in Purview, which is well-known and widely used visualization tool. This is the simulation output, visualized in Purview. Now we can see a protein surface which changes in each time step. In another example are two spheres which interact, as well as, at previous example, colors of the protein represents internal forces in each node. Surface of each protein is divided into triangles, which are divided into another 4 triangles. These blue triangles are base of tetrahedrals, which are the basic building blocks of each protein. We can see them if we add internal lines. Now we focus on one blob. Arrows from each node represent strength and also direction of the internal force. We may also observe values of the force of selected nodes or set a threshold. As I mentioned earlier, proteins are usually known in their atomic structure, which is stored in PDB, Protein Data Bank file format. As you may see, it is also possible to visualize them in Purview. PDB file is at the beginning of the simulation when it is discretized into alternative representation. Because Purview can visualize both the VTK and PDB files, it is also possible to do PDB-VTK mapping, which gives us a really important point of view.